Hey, first hour, how's it going? What's new? Give me an update. Have you learned any new skills or been creative in any way? I think I'm going to try my hand at making masks after this today. So we're going to talk about Unit 3, Cellular Energetics. And I think as soon as you hear energetics, you're thinking of enzymes, you're thinking of photosynthesis and cellular respiration. So I think the big thing is being able to explain how organisms obtain, transfer, and use their energy. Let's see, how we interact with our environment and how fundamental processes work at the cellular level, so looking for connections. So we're going to talk about the structure and function of enzymes, identify some of the roles of energy in living systems. We'll slightly review photosynthesis, cellular respiration, and we're going to just touch on molecular diversity. And again, this isn't meant to be a total reteaching, just a reminder of the kind of things that you should be studying. Okay, so the big idea, the highly complex organization of living things requires constant input of energy and the exchange of macromolecules. So that's going to be the underlying theme for almost all of these slides. First of all, we need to be able to describe the properties of enzymes, and remember, describe means to pretty much make a mental picture in words. So you would talk about, maybe you would talk about the protein structure, primary, secondary, tertiary, quaternary, and the overall structure of the enzyme is at that quaternary level, right? And it has a specific shape, and that allows it to do a specific job. So you would talk about the active site having that specific shape and matching a specific substrate. In some cases, we have that competition where two molecules fit the same active site because they have similar shapes, and that's going to create some competition. We should be able to explain how enzymes affect the rate of biological reactions. So remember, they're catalysts, they're speeding up chemical reactions. So how is it they're able to speed up chemical reactions? Because they're lowering the activation site. So here was the stereotypical example where you have an uncatalyzed reaction that requires a lot of energy and a catalyzed reaction that requires less. In either case, the reactants start with the same amount of energy and the product ends with the same amount of energy. So the big thing that allows them to lower that activation energy is the positioning of molecules. So if you're doing an anabolic or synthesis reaction, you have two molecules that may or may not come into contact with each other. But if there's an enzyme that holds the two of them in place, and then when they have that conformational change that enhances it, it puts those two in proper orientation to create that bond. And so that's going to lower the activation energy. The same thing, similar, if it's a decomposition reaction or a catabolic reaction, you have the one molecule that's fit with the substrate, or the enzyme, and then when the enzyme changes its shape, it will weaken the bonds that are within that substrate, and so it allows that substrate to break apart more easily. So I think the big thing is about positioning. Explain how changes in the structure of the enzyme may affect its function. So when you're talking about changes in structure, you're probably thinking right away about denaturation. So we've talked about how temperature, pH, and salinity can affect the tertiary and quaternary levels of proteins. And since enzymes are protein, anything that disrupts a protein will disrupt an enzyme. So pH, remember, is an inverse log of the hydrogen ion concentration. So the more hydrogen ions there are, you can see that that would very easily disrupt the hydrogen bonding that occurs within the primary and the secondary structure of the protein, and if it affects the secondary, it's going to affect the tertiary and the quaternary that follows. Ionic interactions, so salinity would fall into this factor. A lot of those side chains, right, they're either positive or negative, and so an ionic interaction in the tertiary level can very easily be affected by salinity, changing the shape of that function and therefore, or the shape of the molecule. And if the molecule can no longer fit the structure, then it can no longer do its job. Okay, so effect of the environment basically is what we're looking at here. So pH, temperature, and substrate concentration. We know that enzymes work at an optimal pH, optimal temperature. So on these graphs, why is this one raising, right? It's getting closer and closer to its optimal pH, so it's going to be able to have more reactions, and at a certain point, it's going to denature and it falls apart. With temperature, it's a little bit different. It has more to do with the kinetic energy of molecules. So we know as we heat up, molecules move more quickly, and if they're moving more quickly, they're more likely to collide and therefore create interactions, right? Create bonding. And then as at some point it gets too hot and the hydrogen bonds in the protein are disrupted, and then that will denature the molecule altogether. You can refer to the substrate concentration or the enzyme concentration. It just makes sense. The more of them there are, the greater rate the reaction can have, because there's more chances for collisions. At some point, all your active sites will be filled up. Maybe that's your substrate level saturation point, right? All of the active sites are filled with substrate. It can't go any faster than that. Or once all the substrate has been converted to product, it can't go any faster than that. Some other things that could be in the environment are other molecules that either compete or inhibit. So we have competition where molecules might be filling the substrate's active site, so the substrate can't bind. Or we have our non-competitive inhibitors where they bind at an allosteric, allo other. So they can bind at an allosteric site, and when that happens, there's a conformational change. So this active site will no longer fit the substrate. Describe the role of energy in living things. So a lot of times kids answer free response questions at the beginning of the year, and they don't really know how to express the importance of energy. They just want to connect energy with life, and all living things require energy, and if they don't have energy, they'll die. But why would they die? What do they need their energy for? So be very specific. Tell the readers what you know. So some of the things that we use energy for, cell respiration itself requires energy. So it requires energy to break down food in order to release more energy. Cell reproduction requires energy. Active transport, that sodium-potassium pump. Movement of molecules within the cell or outside of the cell, endocytosis, exocytosis, those require energy. So try to give some specific examples of what the organism would use the energy for. We know that the input of energy must exceed the output of the energy, or there'll be a breakdown, right? If you don't have enough energy for your processes, then there's going to be some catabolism of cells. The metabolic pathways, remember, those are coupled reactions. It's a series of pathways. Usually the energy created from one powers the next reaction. So an exothermic or an exergonic and an endergonic reaction. An exergonic will power the endergonic. You can also talk about the products in the reactant. So the products of one are the reactants of the other. So over here we see glucose is a product, and then it's a reactant down here. So those would be coupled reactions. And again, this energy piece, a lot of that is photosynthesis and cellular respiration. And we know that those are series of reactions with a lot of intermediates along the way. We don't have to know our intermediates, but we can recognize that the molecule is being changed little by little, and that like each reaction, the product becomes the reactant for the next. Describe the photosynthetic process that allows organisms to capture and store energy. So I'm seeing capture, so that's going to make me think of the photosystems, which are doing the job. It says describe, so I'm going to think about creating a picture of those photosystems, perhaps clusters of photosynthetic pigments within the chloroplast, and more specifically within the thylakoid membrane of the chloroplast that is part of the grana, which is part of the chloroplast. So you can also look at levels of organization in that description as well. Remember, all living things need to use energy. So even those first bacteria, they had to capture and use energy as well. Cyanobacteria were the first that we know of that were capturing that energy and using it, and that's where the oxygen in our atmosphere is believed to have come from. So they changed the whole atmosphere, and so all living things adapted after that. So those light-dependent reactions, as you're discussing those photosystems, you would talk about the ATP and the NADPH, because we're saying up here, describe how they capture it and store it. So where's that energy stored? So first it's going to be stored in the form of ATP and NADPH, right? It'll later be transferred and stored in other organic molecules. Okay, so now we're obtaining energy from macromolecules. So this makes me think of cellular respiration. So this says explain how. So now I want to just talk about the process, explaining it to somebody else so that they can understand the biological principles and concepts involved with the process. So I would talk about glycolysis. Well, this whole thing is a series of biological pathways, right? So you might include that. The glycolysis occurs in the cytosol or cytoplasm of the cell, and you're breaking glucose down into pyruvate. And in the process, you're going to make ATP and NADPH. Oh, the P is for photosynthesis. NADH from glucose. So remember we said some processes require energy. So remember, glycolysis did require 2 ATPs to get started. So that's a good example of how our cells need energy. So if you couldn't even start glycolysis, you couldn't capture any of the energy that was in the glucose molecule, right? Because glycolysis is going to precede either aerobic or anaerobic respiration. Okay. From there, ATP and NADH are going to be moving into an electron transport chain, but first let's take that pyruvate and further oxidize it in the mitochondria. So moving into the mitochondria, pyruvate is oxidized one step at a time, and with each conversion, carbon is released in the form of CO2. This is going to produce even more NADH and FADH2, and those are all taking the electrons that were originally in the glucose molecule and moving into the cristae, the inner folds of the mitochondria, into the electron transport chain. So this electron transport chain, chemiosmosis, we talk about that, is going to ultimately make ATP. And if you're in cellular respiration, that process is called oxidative phosphorylation. If you were talking about photosynthesis, this would be substrate level phosphorylation. So NADH and FADH2 are moving into, they're delivering electrons to this electron transport chain. NADH moves in at a lower location, I guess you would say. It powers 3 pumps, whereas FADH2 is closer to the oxygen molecule, so a little bit higher level, and only powers 2 of those pumps. All of these pumps are generating a electrochemical gradient in the form of protons. So they're pumping hydrogens into a high concentration area, building up the gradient so that it'll continue to diffuse through ATP synthase. You don't need to know the molecule names, all of the transport protein. You do need to know ATP synthase, so be familiar with that. Okay, how they capture energy from light and transfer it. So we've kind of already talked about capturing light. You might go a little bit more into the photosystem concept and how electrons are jumping from one photosystem to the next, or one chlorophyll molecule to the next, till they get to a reaction center where the electrons are boosted to a higher energy level. And that's when they move into that electron transport chain where they lose energy to the electron pumps, the hydrogen ion pumps. And then the first, which is photosystem 2, will donate electrons to photosystem 1. Photosystem 1 electrons will end up in the form of NADPH to be used in the electron transport chain. Along the way, this electron transport chain is, that's NADH is going into the Calvin cycle. This electron transport chain is making ATP very much the same way as it occurred in cellular respiration. In the Calvin cycle, again, you have a number of intermediates along the way. You don't need to know all of them. You don't need to know the numbers. Just know that like this is that transfer of energy. So the ATP and NADPH that were made in the light reactions are now being used in the light independent reactions. In the end, this is the step that we make organic molecules. We originally learned we're making glucose, but now we know that those 3 carbons, G3P, can be made into all kinds of different things, right? We might use them to make hydrocarbons and lipids. We might use them to make nucleic acids or to make proteins. So we need to broaden our concept of what organic molecules are made. Okay, now let's use the energy stored in biological macromolecules. So using energy in the molecules, again, you're thinking of cellular respiration, glycolysis, also fermentation. So glycolysis precedes either fermentation or cell respiration. Fermentation might make lactic acid or it might make alcohol, depending on the type of organism that's undergoing it. So like yeast are going to make alcohol, but animals are going to make lactic acid. Only the alcohol fermentation releases carbon dioxide, and lactic acid does not. So neither one of these are producing any more energy. The only energy available is the energy coming from glycolysis. So it's really inefficient. What's the purpose of fermentation? The purpose is to recycle NAD+, because without NAD+, glycolysis cannot continue. Okay, and we've talked about the cell respiration, then the aerobic organisms. And remember, like, not just animals go through cellular respiration, which is a misconception. Every single organism on Earth needs energy. So all organisms go through cellular respiration. Even aerobic bacteria can go through cell respiration. It's just their electron transport chain is going to be embedded in their plasma membrane rather than in a mitochondria. Okay, so I feel like we've talked about that. One other thing about this, entropy, right? The universe tends towards disorder. It requires energy to keep things orderly. So that's another reason why we need energy, because clearly our cells have an order to their structures, right? Cellular respiration loses some of that energy because we know that energy cannot be converted without losing some usable energy. So here's a good place where energy is lost in the form of heat. Okay, this is making a connection to evolution, and that's why you should watch those Unit 7 videos, even though evolution is not on the test. So where can we see variation within organisms as it relates to energy transfer? So clearly aerobic and anaerobic respiration. If an organism has the enzymes available to undergo aerobic respiration, it has that advantage, and if it doesn't, it'll need to go through the anaerobic pathways. We've talked about KMC3, C4 plants. So they all have different ways of dealing with their environment. So that's probably a form of natural selection, right? If you were able to hold onto your water, you had a better chance of surviving in a dry environment than if you couldn't. Chlorophyll A and B, as well as the accessory pigments, allow plants to obtain and be as efficient as possible different wavelengths of light. So different areas of the earth would have different angles of light hitting them, and so they would need a variation to be able to withstand their environment. Types of hemoglobin molecules allowed all different organisms to carry oxygen. We can look at hemoglobin molecules with BLAST and analyze the genetic codes, or the amino acid sequence, I should say, to see how closely related organisms are evolutionary-wise. Also going with this, so we're looking at how molecules within the cells affect the ability of organisms to survive in their environments or reproduce. Also membrane receptor proteins, right? Because those membrane receptor proteins might be receiving signals, ligands, that turn different processes on within the cell, including cell respiration. I wanted to say cell reproduction. Okay, some keywords. We've been talking about keywords. We've already talked about explain and describe, right? So other skills you should be able to do: provide reasoning to justify a claim by connecting evidence to biological theories. So the claim is not enough. That's why we've been doing claim evidence reasoning all along. So make the claim, justify it with evidence. What examples can you give, or what data supports your claim, and then provide reasoning. So why is that piece of evidence indeed evidence? How is it connected? Support a claim with evidence from biological principles, concepts, processes, or data. You are very likely to see this on your test this year. You'll be giving some sort of experiment that you will have to analyze the data for, and make claims that you can support with evidence from the data that is presented. Predict cause and effect of change. So the AP is really big on taking a concept that you already understand and then throwing a wrench in it. If this isn't working, or if this is not shaped correctly. So they like to change it up so that you can use the information that you already know and apply it to a new situation. So if you disrupt it, if carbon wasn't available, if oxygen wasn't available, if you were in a high temperature, if you were in a low salinity, like think of all the ways you could change a system. One or more components in a biological system based on data. Okay, identify experimental procedures. So identify, you're just simply listing. Think about the labs you've done. You've done the cellular respiration lab with the peas. You've done photosynthesis lab with the leaf chads. We've done chromatography. Think of all of those different labs that we've done, and you might be able to make connections to those. So what were the procedures that you needed? Like, why did we use KOH in the cellular respiration lab? I would go back and I would review the components of each of those labs. Identify and justify appropriate controls. So what would your control be in this situation, and why? So you're justifying why would that be a control. Remember, a lot of times we use positive controls and negative controls. Control in any source or any fashion is used as a comparison. If it's a negative control, it's showing you what would happen if the experimental factor had no effect on the system. So the negative control doesn't have the experimental factor. A positive control would show you what you would expect if something was present. So like, if I wanted to test for the presence of glucose and I used Benedict's solution, I would have a positive control that included the Benedict's and the glucose. And then I would see the color it changes, and I can use that to compare to other experiments and see if glucose was present in the other setups. Representing and describing data, construct a graph, plot, or chart. I think this is one that would often show up. I don't think they will ask you to construct a graph, plot, or chart information this year, where you have to upload your information into a computer or a telephone or type your information in. So I don't think you'll be graphing. That's just me. So remember what all of these words mean so that you use them appropriately. In the last unit, we covered describe and explain. So identify is as simple as you can get. It's name, list, and give an example. You don't have to go into a lot of detail behind that. So that should be a very short, quick answer, but answer in the form of a complete sentence, not a bullet and not an outline. Predict. You're saying something that will happen in the future. So if you turn the temperature up, what do you think will happen? You're making a prediction. Effect, a result or a consequence that can be positive or negative. So the effect of using radioactive isotopes, for example. If you used radioactive nitrogen in a soil sample, where would you expect that nitrogen to go? Analyze, to study something closely and carefully. You will have to do this on this year's test. So they will give you information, they will give you experimental results. You need to analyze them. So you're looking for trends, you're identifying trends, and you're going to identify what in the data source allowed you to come up with those trends. Support, back it up, or prove your answer with details. So that would be taking what you read in the prompt, probably in the sample data, for example, and use that to support your claims. Okay, so that's 3 units down. It's technically spring break. We're going to pick up next week, and we're going to review units 1, 2, and 3 that here in Michigan we couldn't get online with our students yet. So we're going to review those together next week when we're finally allowed to get together. And then we'll move forward with units 4, 5, and 6. And then we'll have a week to do some practice with FRQs before our big test on May 18th. Okay, so I hope that was helpful. I hope you guys are all staying safe, keeping your distance. My mom is having a really hard time understanding the concept of social distancing. So hopefully you kids are doing a better job at that. So take care. I hope to see you soon. Bye-bye.